Good evening, ladies and gentlemen. My name is Mohanraj Krishnan, and today I'm going to be presenting my PhD project, which is investigating the genetic causes of childhood obesity using the Children of Scope cohort, which is an Auckland-led study consisting of 6-year-old children. I'll be also looking at environmental factors and how this may influence or modify the genetic risk of obesity in these children. As most of you know, obesity is a serious public health issue and is associated with long-term increased risk of diabetes, heart disease, and early death. More alarmingly though, children are becoming more obese and they're presenting with the same clinical symptoms. And it's no different in New Zealand, with approximately 1 in 3 children either overweight or obese. Furthermore, within New Zealand, there are ethnic differences, with the Maori and Pacific Island children showing higher rates of obesity when compared to European children. Now, there are a number of factors that can contribute to the development of obesity, and this ranges from what the mother experiences during pregnancy to the ever-growing surplus environment and to the microbiology of the gut. But for today's talk, I'm going to give you a genetics perspective and how this may contribute to the development of obesity. But why is obesity such a major issue in today's society? The thrifty gene hypothesis was proposed to explain the modern emergence of obesity. Historically, obesity was considered an advantageous trait during times of feast and famine. During feast, early humans would store fat in preparation for famine. Now, those individuals that had the genetic tendency to store and utilize fat more efficiently were likely to survive famine and pass on their genetics to subsequent generations. Similarly, these humans went through activity and rest cycles, and those that were physically capable of hunting and gathering during famine were likely to survive. However, in today's society, there is no famine. Instead, it's replaced by the constant availability of food and decreased exercise levels. This creates a mismatch between our genes that have evolved to protect us with today's surplus environment, resulting in excessive fat storage and obesity. However, there are a few fundamental flaws associated with this very simple concept, mostly regarding the timing, the strength, and frequency of famine as a selection driver. The thrifty gene hypothesis states there was no selection pressure of famine on our genes. It remained neutral, and we randomly accumulated these mutations or spelling mistakes in our DNA code over time, and when exposed to a certain environmental condition, such as today's society, those unfortunate to have the wrong combination of spelling at increased risk of becoming obese. More recently, though, it has been suggested that the presence of the gene is not enough. It's how a gene works and when it works which is more important. The thrifty phenotype hypothesis proposes how a baby develops during pregnancy is based on what the mother experiences in her own environment. For example, if a mother is exposed to a poor nutrient environment, certain genes will be switched on and off to promote and store fat for that baby to increase its survival chance for that expected environment. Once again, if there's a mismatch between that environment and the genes that have been switched on and off, then there is increased risk of disease and obesity. My project is looking at certain aspects of the thrifty gene hypothesis. More specifically, I aim to identify genes and its association with obesity traits, BMI, and percentage body fat in New Zealand children. And we know that these genes interact with the environment to contribute to the development of obesity, and therefore I'll be looking at environmental factors, in particular diet and exercise, and how this may modify the genetic risk of obesity. Our study consists of approximately 1,200 6-year-old children in which we collected genetic information from them from either blood or saliva. We selected approximately 80 genes, and these were measured using a technology which detects these spelling mistakes, as shown in this diagram here. Each dot here represents an individual, and each cluster represents a specific combination of spelling. What we did is to compare the frequency between these combinations and determine whether there is a significant difference between them in explaining obesity risk in these children. So here are 2 examples of genes associated with BMI found in our study. What we want to know is what effect these variations of spelling has on BMI scores. So here are the BMI scores for all the children having the T spelling of the NEGR1 gene. As you can see, children having the C version of the same gene showed a small shift to the left, indicating a protective effect. Similarly, children having the A version of the OLFM4 gene showed a small shift to the right, indicating a susceptible effect. I want to stress the point that these genes have varying effects, but it's usually a small one. This means something else is driving the obesity problem. We found many genes associated with obesity traits across many different biological pathways, and this includes appetite regulation, storage of fat, sleep, stress. We also found two different subsets of genes associated with BMI and percentage body fat, with only a small sub-overlap between them. We think we are measuring association for that particular obesity trait and not overall clinical obesity. Using other indicators of obesity, such as waist circumference and waist to hip ratio, may help us gather more information and give us a better picture on how obesity develops and identify genes associated with overall clinical obesity and not just for that particular trait. Alongside with the genetics, I've been looking at environmental factors and how they can contribute to the involvement of obesity. For example, I looked at mother's weight during pregnancy and how that may influence a child's weight gain later on in life. But for today's talk, I'm going to be presenting the results of dietary habits of these children and how different diets can interact with genes to increase risk of obesity. I'll be also talking about physical activity and how this may modify the genetic risk of obesity in these children. Dietary information was collected from these children using food frequency questionnaires and separated into three broad categories: a healthy diet, a traditional diet, and a junk diet. And the independent score was given for each child for that particular diet. Here's an example of a healthy diet interaction with the RSPO3 gene, which is known to regulate appetite. Here are the BMI scores for all three combinations from the RSPO3 gene. As you can see, there is massive overlap between each one, indicating there is no difference in BMI score, which means there is no interaction between this gene and this diet. We observed a similar pattern with a traditional diet and the same gene. But when the same gene is exposed to a junk diet, the GG combination showed higher BMI scores. So children having this combination, when exposed to a junk diet, are more susceptible to having higher BMI, indicating a junk diet gene interaction. We have also collected 1 week's worth of physical activity from these children using these devices called accelerometers, which converts acceleration of the body into activity counts. Here's an example actograph, which is an output file of these accelerometers over a 1-day period. Each peak here represents intensity of exercise over 1-minute time intervals. From this data, we also can determine sleep patterns from these children, as shown by the relative lack of inactivity around these times. Using this, we can determine whether differences in sleep and intensity of exercise is directly correlated to differences in obese outcomes in these children. This study offers us a great opportunity to further our understanding between the genes and the environment and how they interact with each other to contribute to the development of obesity in the context of early life development. And although we do have this fixed genetic risk, intervening early in life by modifying certain components of an environment gives us a better outcome in reducing obesity when compared to intervening later on in adulthood. I'd like to thank my supervisors, Professor Andrew Schelling, Leslie McCowan, and Associate Professor John Thompson. I'd like to thank my advisor, Dr. Lindsay Cree, and the collaborators and participants of the Children of Scope study. I'd also like to thank the members of the Schelling and Cree Lab and my funding bodies, Gravita and HRC. And I'd also like to thank you for listening to me give my talk today. Thank you.
When you identified specific genes, and you took you developed the statistical analysis of particular individuals, did you isolate other variables which could be influential in the outcome? So, for example, did a certain cultural group or a certain people group have a higher instance of a certain gene, and therefore, or maybe more males have a certain?
That is a good question. Our ethnicity is a major issue in obesity. We know certain ethnicities have higher risk of obesity. For example, like I mentioned, the Maori and Pacific Island population, and even the Pima Indians in the North American. All of them have the same gene, but all of them have different variations of the gene. The Maori population may have one different combination of the gene which increases that risk of obesity, and that's what we're looking at. We're looking at the presence of what kind of variations do they have in that gene that makes them more susceptible of becoming obese.
But my question is it not possible to find those genes in a European? I'm sure that there is, because we are able to have crossed, we're the same.
Yeah, there is the same genes in the European population, but the fact is, this gene, I've got to make sure.
Did you sample from all the different?
No, we didn't sample. Our cohort consists of mostly Caucasian children. We've got mostly our children from the Auckland hospital, and which had a 95% of European ethnicity. We do have some Maori, Asian, and Pacific Island population, but one of the problems with these studies is power. If you don't have enough population, you decrease the power, and you may have an increased association and discover false discovery genes. So you might find an association, but is that true? Because you only have such a small sample set from those populations. So we cannot really break it down on ethnicity based on the fact 95% of our population is of European descent. But in saying that, we are collecting samples from Middlemore, which are mostly have a higher percentage of Maori and Pacific Island children. We can access cohorts from Finland and Norway, which have really good detailed cohorts. We would collaborate with England and Ireland also, which have a different European ethnicity there. So adding all of this can increase power and also maybe determine if geographical location also have differences in the frequency of having different variations in those genes as well.
Could be something outside that's isolated, like for instance, could it be an additive in the food or the water which actually triggers it off?
Well, for this project, we've been looking at the gene, and the genes could interact with certain components of food that may increase risk of obesity. For example, we know that certain variations of genes can interact with higher fatty foods that may increase or decrease risk of obesity with them. In terms of additive of stuff in water and food, yeah, there may be a possibility. I did not specifically investigate certain components of that, but we have looked at dietary patterns of these children. We have looked at breastfeeding from the mother, and all of these, could be included. We've looked at smoking status of these mothers as well. But in saying that, we have a low frequency of mothers that smoke, because a lot of published literature knows nowadays that smoking is hazardous during pregnancy, and so a lot of the mothers don't smoke. But we have a great biobank which we collected a lot of information, even pregnancy data from 15 weeks, 20 weeks, and 72 weeks. And we collected hormones from these mothers as well. So we can correlate all of this with the genes to see if they have increased or decreased risk of obesity. So with the dietary stuff, I've just showed dietary today, but we've broken down into 3 patterns. So yeah, there could be possible association between what's in the environment and how they interact with the genes to increase obesity risk, in particular ethnicities as well.
Hi, have you looked at any correlation between poverty and
Yes.
these genetic markers?
We do have socioeconomic class, and we also have education levels of the parents as well.
Because you can look at junk diets and they're a lot cheaper
Yes.
than sort of healthy diets.
Exactly.
And therefore poverty is obviously effective.
And that's a good start. We can first of all break it down into looking at whether socioeconomic class and poverty in these populations associated with higher increase of junk diet. we can look at that first and say, okay, there is a correlation between poverty and junk diet. And then we can go one step further and say, is it a transition from genes with poverty, or is it mediated by the fact it's the junk diet based on poverty? So we can look at that model as well. And we will be looking at that and creating a profile, as you can say, with how environment and certain components of environment interact with the genes to increase obesity risk in children, and hopefully translate this into adults as well.